हेलो बच्चों, वेलकम टू SPSVERSITY. In the previous session, we discussed about the plant growth and development. So, growth is a part of development. इसलिए ये ग्रोथ और डेवलपमेंट के लिए क्या चाहिए, इसको क्या बोलते हैं कि प्लांट ग्रोथ रेगुलेटर्स और plant hormones. So what is meant by plant growth regulator? So plant growth regulators are chemical substances that influence the growth and differentiation of plants. It will influence the growth and differentiation of plant cells. And they act like a chemical messenger, so which will coordinate to each other. So this chemical messenger will promote the coordination of all the cells. So here plant growth regulators or chemical substances that influence the growth, so which will do intercellular communication. Phytohormones. Phyto means the hormones which are secreted in plants. Phyto means plants. So इसको क्या बोलते हैं कि phytohormones. कुछ hormones will promote the growth. So इसको क्या बोलते हैं, growth promoters. कुछ हार्मोन्स they will inhibit, इसको क्या बोलते हैं कि growth inhibitors. So which promote growth, उसको क्या बोलते हैं कि growth promoters. Which inhibit, they are called growth inhibitors. Growth promoters mean auxins, gibberellins, cytokinins. Growth inhibitors में abscisic acid, ethylene. There are majorly 5 phytohormones: auxins, gibberellins, cytokinins, abscisic acid, ethylene. ये ऑक्सिन्स, जिबरलिन्स, साइटोकाइनिन्स ग्रोथ प्रोमोटर्स में आता है. एब्सिसिक एसिड और इथिलीन ग्रोथ इनहिबिटर्स में आता है. पहले हम डिस्कस करेंगे ऑक्सिन्स की बारे में. तो these auxins naturally they are present in the form of, in plants, if you take, they are called IAA, IBA. Indole acetic acid, indole butyric acid. And there are synthetic hormones which are synthesized commercially. That is 2,4-D naphthalene acetic acid. 2,4-D or 2,4,5-T, ये पूरा synthetic hormones. Synthetic means which are synthesized artificially. When you take IAA, IBA, they are naturally present in the plants. पहले auxins were isolated from plants, that is by F.W. Wendt. And he has isolated from the tip of coleoptiles. Coleoptile means which is the protective covering of plumule. So in embryo, the plumule is protected by a cap-like structure. इसको क्या बोलते हैं कि coleoptile. वो coleoptile से auxins को isolate किया. And when you come to the discovery of auxins, it is done by Charles Darwin and his son Francis Darwin. They were discovered by Charles Darwin and Francis Darwin. Okay. So here if you take this auxins, so generally auxins are synthesized in the apices. The apices means root apex, shoot apex, branch apex. So here you can see. So this is the coleoptile, the apex of the coleoptile, where auxins are synthesized. So these are auxin molecules. So here if you look into this, the unilateral light, when light falls on one side of this coleoptile. So there is less distribution of auxins on this side where light falls. On the shaded region, there is high distribution of this. Okay. Due to this, what happens is the more cell elongation takes place on this side, which leads to bending of coleoptile towards the source of light. So here you can see, when coleoptile is exposed to unilateral light means it is exposed to only on one side. So here what happens is, towards the sunlight side, it shows that the distribution will stop. And you can see the lateral distribution of auxins on the shaded region. Okay. So due to high concentration of auxins, what happens is there is more rapid growth on the shaded region, resulting in a curve just towards light. Okay. So growth will proceed in proportion to auxin concentration. So this is how the auxins will play a role in the physiology of plants. Okay. So what are the functions of auxins? Auxins will promote apical dominance. What is meant by apical dominance? Due to presence of apical bud, the axillary bud growth is inhibited. Okay. Due to presence of apical bud, the axillary bud growth is inhibited. It is called apical dominance. Due to this, the plant will grow taller. To make this shorter, what gardeners will do is they will cut this apical bud. Initiate rooting in stem cutting. So during tissue culture to promote root formation, and in horticulture this grafting to promote rooting, they will use auxins. Prevent fruit and leaf drop at early stage. Induce parthenocarpy. Parthenocarpy means formation of fruits without fertilization. So formation of fruits, fertilization के बिना उसको क्या बोलते हैं कि parthenocarpy. Promote abscission of older mature leaves and fruits. Abscission means a layer of dead layer is formed between the plant, the leaves and fruits, so that they will fall down when they reach to maturity. And they will work as weedicides. Weedicides means which will kill weeds. So weeds means the unwanted plants present in the field. ये monocot crop plants में use करते. So these are the functions of auxins. Okay. So auxins is one growth promoter. And when you come to gibberellins, so these are first identified by a Japanese scientist, उसका नाम Kurosawa, and he has identified in a fungal pathogen, that is Gibberella fujikuri, okay, which will cause disease in rice seedlings. Okay. And it was extracted from this rice seedlings, which will cause Buckney disease. Due to this, the stem will elongation, we can see abnormal elongation. Okay. The gibberellins में बहुत सारे types है, like gibberellin 1, gibberellin 2, gibberellin 3, इसको क्या बोलते हैं कि types of gibberellins. It is present in a fungi that is called Gibberella fujikuri. Okay. A functions of gibberellins को लियो तो, so पहला function है कि elongation of stem, that is the stem shows elongation during the growth. Bolting and rosety plants, that is in rosety plants where the leaves are arranged very closely, during this we can see the stem elongation. It will promote seed germination. So during the process, the seed germination, so a lot of energy is required. So it will promote this energy requirement, it will indirectly promote seed germination. Overcome the natural dormancy of buds, tubers and seeds. So dormancy बोले तो resting period. So even though they are provided with suitable conditions, they will not germinate. It is called dormancy, seed dormancy. So seeds require some favorable conditions. Even though they are provided with favorable conditions, they don't germinate. That is called dormancy of buds and tubers and seeds. It can be overcome by gibberellins. It can be overcome by gibberellins. Okay. So अभी तक हमें पढ़ा है ऑक्सिन्स और जिबरलिन्स के बारे में. Now we are going to study about cytokinins, which is another plant growth promoter. It was discovered by Skoog and Miller. Okay. And it is crystallized from corn kernels and coconut milk. ये कोकोनट मिल्क और कॉर्न कर्नल से एक्टिव सब्सटेंस, इसको क्या बोलते हैं कि काइनेटिन, so which is extracted from this corn kernels and coconut. So why the name cytokinins has come? The reason is it will promote cytokinesis during cell division. Cell division में division of cytoplasm को क्या बोलते हैं कि cytokinesis. So cell division में interphase M phase रहता और M phase में karyokinesis cytokinesis रहता. Cytokinesis is division of cytoplasm. उसको a cytokinins, it will promote this one, इसलिए उसका नाम cytokinins रखा. इसमें functions of cytokinins लिए तो पहला function delay of senescence. Senescence means falling of leaves. which leads to death of leaves and flowers, which is delayed. The process is delayed. It promotes lateral budding, formation of lateral buds. It induces cell division, particularly when you come to the process. It is an active promoting substance of cytokinesis. So cytokinesis is promoted by this cytokinins. So cytokinins का functions: delay of senescence, promotion of lateral budding, inducing of cell division. So ये functions, and at the same time it will promote the opening of stomata. So ये functions of cytokinins. So अभी तक हमें पढ़ाया ग्रोथ प्रमोटर्स के बारे में, ऑक्सिन्स, जिबरलिन्स, cytokinins. Follow us on social media, that is SPSVERSITY. ठीक है, अभी हमें पढ़ेंगे ग्रोथ इनहिबिटर्स के बारे में. इसमें आता है कि दो, abscisic acid और ethylene. So abscisic acid, it is identified by Adicott, Frederick T. Adicott, and the name abscisic acid, it is come from the term abscission. It was isolated from several abscission exudating substances from cotton plants. And it is also called stress hormone. During stress conditions, it is secreted. अब देख सकते हैं पिक्चर में, which will promote the closing of stomata. Okay. Due to closing of stomata, the transpiration rate is controlled. Okay. इसलिए इधर क्या होता है कि abscisic acid binding will leads to influx of calcium. Influx of calcium means movement of calcium inside, and opening of K+, which will be K+ will be sent out. Due to this, what happens is water is lost from God cells, which leads to closing of stomata. So it will promote closing of stomata. Whenever there is a water less during this stress conditions, it will promote the closing of stomata. Okay. Basically, when we take abscisic acid, it is a plant growth inhibitor. It will induce seed and bud dormancy. Okay. ज़्यादा होता है seed और bud dormancy. It will inhibit seed germination. It will stimulate the closure of stomata, and moreover, it is a stress hormone. So, ये functions of abscisic acid, which is a growth inhibitor. Okay. And when you come to ethylene, which is a ripening hormone, and this is the only gaseous hormone. Whatever you have studied in all these hormones, ethylene is the gaseous hormone, which is volatile. It is called fruit ripening hormone. Generally, from ripened fruits, if you see this, a gaseous substance will be released, that is nothing but ethylene, which will promote the ripening of the unripened fruits. And this volatile substance is the ethylene. अब देख सकते हैं इधर Gradually the fruit ripenings, banana ripening. Not only banana, many fruits. Okay. So here you can see the action of ethylene. So due to this action of ethylene, the fruit ripening takes place. It is called fruit ripening hormone. Okay. When you look into functions of ethylene, basically a growth inhibitor here. So which will inhibit the growth. So this growth inhibitor, if you look into functions, it will promote senescence and abscission, inhibit elongation of stem roots, so that it will promote the lateral growth. So when the elongation is inhibited, it will promote the lateral growth. Induce fruit ripening. As well as it has some, even though it is a growth inhibitor, it has some promoting effects also. Like it will break seed and bud dormancy, which will initiate germination in peanut seeds. Peanut means the individual which will initiate the germination in groundnut seeds. So these are the functions of ethylene. So अभी तक हमें पढ़ा है फाइटो हार्मोन्स की बारे में. So फाइटो हार्मोन्स में लिए तो ऑक्सिन्स, जिबरलिन्स, साइटोकाइनिन्स, एब्सिसिक एसिड, इत्यादि. ठीक. After this, growth hormone promoters and growth inhibitors, we are going to study about photoperiodism. The name itself says photo bole to sunlight. Okay. So sunlight has lot of impact, lot of effect on plants. So daytime में sunlight has lot of influence on us. Okay. It is a response of plants to relative length of dark and light periods. दिन में plants are exposed to sunlight. रात में कोई sunlight नहीं रहता. Okay. So this sunlight will have lot of impact on many physiological functions of the plants. Like when you come to sunlight, it will induce flowering. Okay. Some plants will require exposure to light to induce flowering. Some plants, कुछ plants को dark period is required to induce flowering. Based on this, if you take the length of time to which plants are exposed, like if you take short day plants को क्या बोलते हैं कि दूसरा नाम क्या है कि long night plants. Quite opposite. Long day plants इसको क्या बोलते हैं कि short night plants. You can look into this. This is critical photoperiodic time. Short day plants where they are exposed to light less than the critical length of the light period. In between there is a flash which will, okay. And when you come to long day plants, they are exposed to light more than the critical length. Okay. And when they are given flash in between, it will promote flowering. So based on this photoperiodism, so plants are classified into short day plants, long day plants, neutral day plants. Here you can look into this. Day length less than, that is 12 hours of flowering, that is critical flowering. So day length less than the critical length of 12 hours of flowering. इसको क्या बोलते, लॉन्ग नाइट प्लांट्स. इसको क्या बोलते, लॉन्ग नाइट प्लांट्स. If you take the 24 hours of a day, okay, this very less than 12 hours is exposed to light, which will promote flowering. Long day plants where the day length is more than 12 hours. If it is more than the 12 hours, they are also called short night plants. These are short night plants. These are long night plants. Day neutral plants. So their day length is immaterial, whether they're exposed to 12 hours or whether they're exposed to less than 12 hours of photoperiodism. It is immaterial. They are all affected by the light. इसको क्या बोलते कि day neutral plants. ऐसे में based on the photoperiodism, if you take the classification, short day plants, long day plants, day neutral plants. Short day are also called long night. Long day plants are also called short night. Day neutral plants, they are not affected by any other light effect, whether they are exposed to less than 12 hours later. So photoperiodism is the length of exposure of light to plants to initiate flowering. So photoperiodism is related to flowering of the plants. बाद में हम आएंगे vernalization. So vernalization is the process in which the plants are exposed to low temperature. When they are exposed to low temperature, it initiates flowering. Okay. So here flowering means, the flowering to be done means they should be exposed to the low temperature. And moreover, if you take the definition in other terms also, flowering is promoted by cold treatment. Cold treatment means the low temperature to fully hydrated seeds, or it is to a growing plant. पिक्चर में देख सकते हैं vernalized plant, that means which is exposed to low temperature, where we can see flowering. This is non-vernalized plant. इसमें no chilling. Plants are at ordinary temperature. इसमें chilling कोई नहीं है. Plants are at ordinary temperature. इधर आप देख सकते हैं. This exposed to chill temperature, that is at 5 degrees Celsius for a month, which is planted at ordinary temperature. इसमें आप देख सकते हैं flowering. Okay. Photoperiodism is due to exposure to light, but it is due to temperature. इधर आप देखें तो no chilling, planted at ordinary temperature, non-vernalized plant. A non-vernalized plant में आप इसमें flowering नहीं है. So no flowering in these plants. It is a non-vernalized plants. So whenever plants are exposed to low temperature for a particular period, they'll show flowering. Okay. So temperature means not only in growing plants, even seeds to germinate also. They are given cold treatment. Cold treatment means they are exposed to low temperature. They are exposed to low temperature, that is 0 to 5 degrees. And this low temperature, it may vary from plant to plant. It is not same in all plants. It may be few days to few weeks. And if we take generally seeds, after cold treatment, they are allowed to dry and then they are sown. So vernalization also prepares the plants for flowering. Not only plants, even the seeds to germinate. So we are exposing the plants, that is called stimulus, which is perceived by apical meristers. Which plants may roots or leaves may be the potential sites of vernalization. Okay. Roots or leaves may be the potential sites of vernalization. Like linear of biennies, where the roots or leaves may be the receptors of this cold stimulus. Generally, it is received by apical meristems, which are present in the apex of root, stem, branches. So, vernalization and photoperiodism both will promote the what we call the flowering. Photoperiodism is the related to exposure to light, but here it is related to temperature. Both are meant for flowering, but the stimulus is different. It is cold stimulus, there it is light stimulus. So this is the vernalization which takes place in plants as well as in growing plants and as well as seeds. ठीक है. So अभी तक हमें डिस्कस किया फाइटोहार्मोन्स, फोटोपीरियोडिज्म और नेक्स्ट क्या की vernalization. And we'll discuss the last topic, that is seed dormancy. Dormancy बोले तो resting period. Resting period में the seeds में बहुत कम water रहता, सेल एक्टिविटी बहुत कम रहता, मेटाबॉलिज्म भी बहुत कम रहता. So resting phase of, during this resting phase, the seeds will have very less amount of water. इसमें बहुत कम पानी रहता, इसलिए इसका एक्टिविटी भी बहुत कम रहता, सेल एक्टिविटी और मेटाबॉलिज्म भी बहुत कम रहता. okay. इसमें इसको क्या बोलते कि डोरमेंसी. So to germinate this seed dormancy should be broken. So when they are in seed dormancy, the seeds will not germinate, even though they are provided conditions means proper temperature, oxygen, light. Even though they are provided, it will not germinate. Okay. Dormancy की causes क्या है? Impermeable seed coat. Impermeable means the seed coat will not allow any water or oxygen, which will not allow. Mechanically resistant seeds. Seed coat is very hard, which it cannot be broken and come out. Rudimentary embryo. Rudimentary means underdeveloped embryo. Embryo is incompletely developed. इसके वास्ते भी a dormancy. Dormant embryo. That means embryo is in resting phase. Insufficient development. Embryo में development up to date नहीं रहता. That is insufficient development. ठीक है. How to overcome this? To overcome this, there are many methods, like mechanical methods, where they will keep a scar. They'll just structure the seed coat so that it becomes impermeable. Okay. They're exposed to low temperature. Like this, there are many methods which will break this seed dormancy. Like if you apply gibberellins, they will break the seed dormancy. Like, for example, if you take in peanut seed, the dormancy is broken down by ethylene. Like there are some hormones, even abscisic acid. Due to presence of abscisic acid, also seed dormancy देख सकते. So seed dormancy is broken down by generally gibberellins hormones. When they're exposed to gibberellins, the seed dormancy can be broken. This is one method. There are other methods also, like exposure of seeds to cold temperature, which is we call vanillization. Breaking the seed coat, just a small break in the seed coat, which will allow the seed coat, when the seed coat is very hard and resistant. So which will allow the seed coat the permeability of oxygen, water. So like this, farmers will be doing many methods to overcome this seed dormancy. So this is about the seed dormancy. Once seed dormancy is broken down, it leads to germination of seeds. Seeds germination होता. Seeds germination. So this is about the seed dormancy. So हमें पढ़ा है growth, phytohormones, photoperiodism, vanillization, or dormancy. Thank you all. Follow us on social media. We'll meet again in the next session.